Namaskar. Today we'll study the characteristics of cancer cells. So in continuation to our last class where we discussed about the difference between tumor and cancer, the tumor may be benign or malignant, and these malignant tumors are called as cancers, and they may be either solid tumors like carcinoma, adenocarcinoma, sarcoma, myeloma, or maybe the liquid ones, that is hematological malignancies including lymphoma, leukemia. So as long as the uncontrolled proliferation is there, it is benign, and when it is added with dedifferentiation, invasiveness and metastasis, it becomes malignant, that is, it spreads from one place to another place. So there are four characteristics of cancer cells: uncontrolled proliferation, dedifferentiation, invasiveness, metastasis. Let us discuss one by one. The uncontrolled proliferation, we will discuss it under 5 heads: cell cycle transducers, growth factors, no apoptosis, angiogenesis, telomerase expression. So let us recall the cell cycle, and as we know, there are two phases: interphase and M phase or mitosis. The interphase consists of G1 phase or pre-DNA synthesis, S phase or DNA synthesis, and G2 phase or post-DNA synthesis. So there are two checkpoints, one before the S phase or DNA synthesis phase, and another checkpoint before the mitosis phase or M phase. So we can have either positive regulators of cell cycle or negative regulators of cell cycle. Positive regulators of cell cycle like cyclins and cyclin dependent kinases, they help in moving the cell from G0 phase or resting phase to G1 phase, so that the cell cycle will continue. Whereas in the checkpoints, the negative regulators of cell cycle act, like P53, retinoblastoma gene, cyclin dependent kinase inhibitors like P21, P27, and inhibitors of kinases. So we can have two types of cell cycle transducers, that is positive regulators and negative regulators, and these negative regulators actually act as the tumor suppressors. They function when some abnormal cell is to be produced, so they stop the cell cycle, they stop the cell division, so we call them as tumor suppressor genes. And when these negative regulators are suppressed or inactivated, then only there is uncontrolled proliferation. So normally, in case of abnormal cell division, in these checkpoints, the negative regulators of cell cycle stop the cell cycle, and there will be no abnormal cell division. Whereas when these negative regulators do not function, then the cell cycle continues and we will have uncontrolled proliferation. There are certain genes called oncogenes which are produced on activation of the proto-oncogenes like CIS, ERBB, JUN, FOS, MYC, etc. And activation of these proto-oncogenes to oncogenes also leads to uncontrolled proliferation. So this is the first point, cell cycle transducers. Next we will discuss on growth factors, how growth factors regulate the uncontrolled proliferation. Growth factors actually act on their specific receptors, which are tyrosine kinase linked receptors, and stimulate the cell in G0 phase to enter G1 phase. So here the growth factors act and stimulate the cell to move from G0 to G1. and that stimulate the production of the positive regulators like cyclins and cyclin dependent kinases. So growth factors like epidermal growth factor, insulin like growth factor, platelet derived growth factor, transforming growth factor beta, vascular endothelial growth factor, all of them act on the specific receptors which are tyrosine kinase linked receptors and stimulate the production of positive regulators. And this also leads to uncontrolled proliferation. Then the third point, no apoptosis. Usually in case of abnormal cells there should be apoptosis, which is programmed cell death for disposal of abnormal cells. So apoptosis occurs to dispose the abnormal cells, but uncontrolled proliferation occurs when there is resistance to apoptosis, when there is inactivation of pro-apoptotic factors. Pro-apoptotic factors usually induce the apoptosis, so inactivation of pro-apoptotic factors inhibits apoptosis and hence uncontrolled proliferation. And another cause of uncontrolled proliferation can be activation of anti-apoptotic factors. So all these three leads to no apoptosis and hence there will be uncontrolled proliferation. Then the fourth point is angiogenesis. Angiogenesis, angio means blood vessels, genesis is formation of new blood vessels. So when there is tumor of 1 to 2 millimeter, they receive nutrient through diffusion from the nearby area. But when the tumor size is more than 2 millimeter, due to the action of vascular endothelial growth factors, there will be angiogenesis, that is development of new blood vessels, and the tumor gets nutrients and there will be growth of the tumor. So in cancer cells, because of this angiogenesis, there is uncontrolled proliferation. Then finally we will discuss the uncontrolled proliferation on account of telomerase expression. So what is telomerase? Telomerase is an enzyme which is highly expressed in cancer cells, and this maintains and provides stability to telomeres. Telomere is a protective cap over the distal extremity of a chromosomal arm. So suppose it is the chromosome, and in the chromosomal arm distal end there is telomere, and telomerase maintains and provides stability to this telomere. So actually what happens, after each cell division a portion of telomere gets eroded, and a time comes when this telomere is completely vanished, so the cell will not undergo further cell division. So ultimately it becomes non-functional after a number of cell divisions. So there will be no more cell division. But because of increased expression of telomerase in case of cancer cells, this telomere is maintained and hence it confers immortality to cancer cells, and hence the cell division continues and there is uncontrolled proliferation. So this is all about uncontrolled proliferation, and as long as uncontrolled proliferation is there, there is tumor and that tumor is benign. And when these three, dedifferentiation, invasiveness and metastasis, these three characteristics are added, the benign tumor now becomes malignant or cancer cells. So now let us discuss about the dedifferentiation. So what is differentiation? When a parent cell divides, it forms two daughter cells. These daughter cells, because of differentiation, becomes mature cells and behave like their parent cells, showing the same properties. They carry out the same functions as that of the parent cell. For example, the skeletal muscle. So the skeletal muscle, the new cells formed, the daughter cells formed, will start contracting. That is the function of the parent skeletal muscle fiber. But cancer cells do not differentiate, or we can say poorly differentiated. So because of poor differentiation, cancer cells multiply faster. So when there is well differentiated cancers as compared to the well differentiated cancers, the poorly differentiated cancers multiply faster. So cancer cells, because of no differentiation, the multiplication is faster, and we can say the poorly differentiated cancers multiply faster than well differentiated cancers, so more dangerous. Then the third characteristic is invasiveness. Invasiveness comes from the word invasion. Invasiveness. Normal cells usually not found outside the tissue of origin. They are restricted to their tissues. But due to mutations in cancer cells, they invade nearby organs. For example, if there is rectal cancer, so the cancer cells of rectum can invade the nearby pelvis. So that is invasiveness, and because of this it spreads. The fourth characteristic of cancer cells is metastasis, and metastases are the secondary tumors. So we have a primary tumor, and a part of these primary tumors called metastasis, through either blood vessels or lymphatics, through the blood circulation or lymphatic circulation, reach other sites and grow at the new site. So that is called metastasis, and because of these two, invasiveness and metastasis, the tumor spreads and becomes cancer. So these are all the characteristics of cancer cells.